happy here today to introduce Taylor McElory. She's a fourth-year PhD student in my lab, but she's only been with me for 2.5 years. And during this time, she has gotten a lot of things accomplished. So one thing you should know about Taylor, she's an avid Doctor Who fan. That's one of her passions. She's avid about all the doctors. And when the new season would start, those were conversations every Monday morning about, did you see Doctor Who? What did you think about this? Blah, blah, blah. So it's been a wonderful, wonderful adventure having her here in the lab. So one thing that I like to tell my graduate students is that, doing science is all about giving back to the next generation. And one of the things Taylor was always great about was volunteering and talking to younger students about her science, whether that was volunteering at the Wonder Lab, going to an elementary school to talk about her research. She was all about volunteering. Another thing we talk about a lot is that science is a team. Science is a team sport, right? So, you've got to be part of the team. You're always willing to contribute. And Taylor was always willing to contribute, whether that was with the current students we had in the lab. This is one of the potlucks we had throughout the summer. She was really great at not only helping, but also teaching undergrads about science, always enthusiastic, And the last thing I want to say is that one of the things we talk about in the Analabs is concept of if you don't publish it, it doesn't matter, it doesn't exist, right? So what I want to highlight here is that during the 2 years she's been here, she's managed to get 2 first author publications and also contribute to other publications going on in the lab. And what's really neat about her is that she was always willing to come in, always willing to help students, always willing to learn new things, before she arrived, we had very, very, very baseline work doing proteomics. And that was another arm of research she brought to our lab. So, it's been great having her here during these past 2.5 years. She's gotten multiple postdoc offers, enough to where she was willing to negotiate her salary. So that's a really good thing. And I'm just excited for her to talk about her work today. So without further ado, I want to introduce Ms. Taylor McElory. All right, let me get out of this.
Okay, yeah, I'll have to wait till you stop sharing.
There you go.
Thank you. No. One second. Accept. We stop share real quick. I have to go under. Okay. Can everyone see my PowerPoint?
Mm-hmm.
Perfect. Thank you, Dr. Allen, for that lovely introduction. I'm surprised you didn't call me by my lab nickname, Robin, basically because other people won't know what to call me by or know what you're talking about even. I'm excited to tell you about the past 2.5, 3 years of work I've done looking at the effects of specific redox modifier, MNBME, BUOE on cognition in female mass after breast cancer chemotherapy. Now I'm going to orient you to our presentation by giving a brief outline. First, I'll give you an introduction to our disease of interest, which is breast cancer, and a common chemotherapy regimen used to treat that. Next, we'll go into one of the main portions of my dissertation, which is looking at the effects of breast cancer chemotherapy on mood disorders, such as anxiety and depression. Next, we'll look at the cognitive side effects of breast cancer chemotherapy, and with introduction into this redox modifier that could be used as a potential therapeutic. I will go into, lastly, some possible mechanisms behind this redox modifier, and then end up with some conclusions. Breast cancer is the most common cancer in American women. It has an average risk of 12%, or that means a woman has about 1 in 8 chance in her lifetime of developing this cancer. However, with the increase in earlier diagnoses and the treatment modalities such as radiation, chemotherapy, and surgery, we have seen an increase in survival rates. For example, local breast cancer, its 5-year relative survival rate is 98%, 98 to 99%. And how, if it even goes out to the regionals, to the lymph nodes, we see that go down just to 86%. Breast cancer patients are not unique in that there's an increase in survival, right? We have an estimated number of cancer survivors here on the right in our graph, estimated in the United States from the 1970s to 2040. As you can see, there's an increase in cancer survivors in the millions in all age groups. And this has really opened up a new area of research looking into how we can improve the quality of life of cancer patients, not only during their chemotherapy, but years afterward. Our chemotherapy regimen of interest is called ACT. It's a combination chemotherapy. The A stands for adriamycin, or the generic name is doxorubicin. This is an anthracycline which works by intercalating between the DNA bases and causes DNA double-strand breaks, which ends up causing cell death. Doxorubicin is given in combination with cyclophosphamide. Cyclophosphamide acts also to induce cell death by inhibiting protein synthesis by alkylating DNA and RNA. Then we have our T for our Taxol. Paclitaxel is the generic name for Taxol. This acts through a slightly different mechanism of acting with the cytoskeleton and stabilizing macrotubules to cause cell cycle arrest and cell death. This chemotherapy regimen is used to treat all stages of breast cancer from early to late, and also the 3 different subtypes of breast cancer. So we have our hormone positive, hormone negative, our estrogen receptor positive, negative, and also our triple negative breast cancer. Patients usually receive this chemotherapy in infusions once they have received the AC combination once every 3 weeks for 4 cycles, followed by paclitaxel, which is also given once every 3 weeks for 4 cycles. One of the most common psychiatric comorbidities that happen in breast cancer patients is depression and anxiety. Indeed, this is the breast cancer patients present a unique population with their own psychosocial, medical, and hormonal factors that can all influence their mood. So when we look at depression studies, we really cannot take those from other studies and generalize them to this population. Because of this, there have been some studies done looking at the prevalence of depression and anxiety in breast cancer patients. One study that used the self-rating scale for self-rating anxiety and depression have found that there was an estimated pooled prevalence of 5 to 15% in breast cancer patients. Also want to just note that patients can experience depression and anxiety by themselves or together. However, when looking at longitudinal studies, there tends to be a trend where we see increased depression at earlier stages of the acute phase of treatment, and then that dies off after treatment is over. And for this longitudinal study I mentioned here, this was done by structured diagnostic interviews. Looking at early stage breast cancer patients, they found that 33% of patients were depressed at time of diagnosis. However, that went down to 24% 3 months after diagnosis, which is estimated about halfway through the chemotherapy treatment. That further prevalence went down to 15% at 1 year post-treatment. Given that we see depression rates decline over time and after chemotherapy, it's natural to conclude that chemotherapy could be a potential cofactor in causing depression and anxiety. And of course, there are a multitude of side effects associated with chemotherapy, some of them being pain and fatigue that can influence mood. However, there's also another way that chemotherapy could influence mood, and that is through altering the gut microbiota. The gut microbiota is the assemblage of microorganisms present in a defined environment. And here we're talking about the gastrointestinal tract. The gut microbiota consists of trillions of microorganisms, which include your bacteria, your archaea, fungi, and your viruses. And this creates a complex economic community, which has a big impact on both post-physiological and disease states. So understanding the composition and the functionality of the gut microbiota in response to chemotherapy is key to understanding how it can affect behavior. The microbiome gut-brain axis is a bidirectional communication pathway between the gastrointestinal tract and the central nervous system. We have seen one of the main avenues through which they talk is through the immune system. In altered stress or psychopathology states with depression and behavior, we see It's correlated with disrupted microbiota composition. We see increase in inflammation and also a dysfunction in the intestinal barrier. In human patients, we do see increased levels of inflammation, such as IL-6 and TNF-alpha. Then, interestingly, all this stuff that we see with altered psychopathologies, we see when chemotherapy is given. Specifically, doxorubicin and cyclophosphamide have been associated with intestinal damage, increased inflammation, and lower bacterial diversity. And the inflammation is a key part, since cytokines and pro-inflammatory factors can not only go to talk to the brain directly through the vagus nerve, but also through the peripheral system, going through blood circulation and crossing the blood-brain barrier to actually induce local inflammation. So the goal of my study was to determine if our breast cancer chemotherapy regimen, ACT, will affect anxiety and depressive-like behaviors in our mice, affect intestinal morphology, and our gut microbiota. I'll come to our study design. We start with regular female Black mice, adults at age 12 weeks. We try to recapitulate the dosing schedule for humans, in that we give a combination of doxorubicin and cyclophosphamide once a week for 4 cycles. Then our paclitaxel is started 1 week after our AC, and again, it's given for 4 cycles once a week. All these injections are given via intraperitoneal injections. And we have 3 time points that we're wanting to look at. We have our first time point, which is our AC immediate. So this is where we conduct behavior 24 hours after our last chemotherapy AC injection. And then this will coincide with our halfway mark between our chemotherapy. Then we have our behavior after our ACT immediate. So after the completion of the whole ACT regimen, again, we do behavior 24 hours after the last chemo. Then we have a 30-month or 30-day, 1 month time point, where we let them rest, where we conduct behavior tests 30 days after the last injection. I just want to note that these dosages of chemotherapy we have published and shown that this alters hippocampal morphology and negatively affects cognition. Our behaviors that we use to analyze anxiety and depression are the open field test, tail suspension test, and the forced swim test. So our hypothesis was that given chemotherapy, the gut microbiota would change. So the chemotherapy would perturb the gut microbiota, and I would expect to see this at both immediate time points right after our chemotherapy. However, after 4 weeks, I hypothesized there's a recovery of this gut microbiota back to the normal state, because it's been shown in mice that even when their gut microbiota is disrupted by antibiotic cocktails, for example, they're able to recover their diversity within 1 week. First behavior test we conducted was the open field test to measure anxiety. It measures the willingness of animals to explore, and it's a relatively simple test. It just consists of a box arena, and then using our automatic tracking software, we can predefine areas where we want to track the mice. If you look over here to the right, this is a couple of diagrams of the tracking of a couple of mice from this paper. The wild-type mice, we'll just say they're experiencing they have normal levels of anxiety, and they're more willing to explore the open and brightly lit environment of the open field. However, increased anxiety presents as stigmataxis, or the tendency to spend most of their time around the walls. You can also measure, in addition to locomotor activity, autonomic activity, such as levels of defecation, which can be used to indicate increased anxiety. So first, I want to start out with the first piece of data, looking at my open field results. We use central tendency, or the time spent in the center of the arena, and we look at all 3 time points. My first time point is here with the immediate AC. My chemotherapy group is represented in red. We see no significant changes in our central tendency nor in our fecal count. We move on to our second time point, right after the completion of ACT. We also see no changes in central tendency nor in fecal count. However, at our 1 month time point, we see a significant decrease in central tendency, which indicates increased anxiety. However, we see no changes in fecal count. Next, to measure depressive-like behaviors, we use the tail suspension test and forced swim test. They both act on exposing the mice to short-term inescapable stress, and we measure their response to that. As these tests are aptly named, the tail suspension is where we suspend the mice by their tail for 6 minutes and we watch their activity. The same thing goes for our forced swim test. We put them in a narrow cylinder of water and watch their activity. So normally when you first introduce these animals to this type of stress, they're going to try and escape. What we're measuring is their immobility time, which reflects their willingness to give into behavioral despair. We found that our chemotherapy did not affect any depressive behaviors at all 3 time points. So we have our first immediate time point here, looking at our immobility and our tail suspension and our forced swim test. Again, at our ACT immediate time point, we see no significant differences in immobility time. And at 1 month, we also do not see any significant differences in immobility time. So to summarize, at my AC immediate time point, there were no differences in anxiety or depressive-like behaviors between our normal control and our chemotherapy-treated mice. At our ACT immediate, we also did not see any changes in anxiety or depressive-like behaviors, except 1 month after chemotherapy, we did see a significant increase in anxiety-like behavior, but no changes in depressive behavior. Next, we wanted to look at small intestinal morphology, and a key part of the intestinal barrier is due to the tight junctions. So here we have villi, an example of villi, which are a villus, which are finger-like projections out into the gut lumen, and it serves to increase the surface area for absorption. And then below that we have the crypts, and the crypts are important in that they have a small niche of stem cells that provide all the renewing epithelial cells that make up the villi in the crypt. And there's a relatively high turnover rate of the intestine of 3 to 4 days. When you zoom in, we have these transmembrane proteins that actually help make up the barrier. They're called tight junctions. Claudin and occludin are 2 transmembrane proteins. Then we have an adapter protein, so one that connects to the actin cytoskeleton. And it's been shown that disruptions changes in expression of these proteins are related to changes in gut intestinal permeability. So to look at our tight junction proteins, we used immunohistochemistry. We looked at DAB staining, which DAB is a chromogen, which is activated upon reacting with hydrogen peroxidase, and our secondary antibodies. So we had our primary antibodies against claudin, so 1 and occludin. We took some of the proximal jejunum and sent those off, fixed them overnight and sent them off to the pathology core for staining. We get a counter stain, so we get both our hemoxylin stain and our DAB staining. However, we used image J to deconvolute the image so we can quantify the intensity of the DAB staining. We have 3 different time points again. Our AC immediate, we see a significant decrease in villi and crypt intensities. This has been shown in cyclophosphamide treatment to also induces decreases in claudin expression. For ACT immediate, we still see a significant decrease in claudin in the crypts. However, we see a significant increase in occludin in both the villi and crypts. And we continue to see this increase in occludin in the villi and crypts in the 1 month time group. This is interesting in that both of these transmembrane proteins are important for tight junction, and then we know bacterial metabolites have been shown to increase occludin. But to exactly know what have effect these proteins, these changes, we'd have to revalidate these protein expressions using more quantitative methods such as PCR or Western blotting, and then also to do intestinal permeability assays to see what the functional effect of these changes. Next, we just wanted to look at our gross jejunal morphology, and to that we just did standard H&E staining. The jejunum, if I didn't mention this before, is part of the mouse small intestine. And during our AC immediate, we saw a significant decrease in villi height and crypt depth, and this would be representative here in our images. We have our saline group and we have quite the long villi. However, when we add in our chemotherapy, we see villi blunting or shortening. And we see, it's known with AC treatment, as I said, that induced different intestinal morphology, and that includes decreased villi and decreased crypt depth. At our ACT immediate time group after a paclitaxel, we can see a continued decrease in villi height. And using paclitaxel as a single agent has been shown to decrease villi length in the intestine. At our 1 month time point, we see a significant decrease in depth. So it's interesting to see what effect that these chemotherapies are having on the stem cell properties of the crypt, in the crypt. However, we need to investigate that more. Next, we want to take a census of the gut microbiota, or basically seeing what's there and how much abundance. First, I collected the fecal samples from our mice. We froze them in liquid nitrogen. Then we sent them to RTL Genomics for DNA extraction, the library prep, and sequencing for 16S ribosomal RNA sequencing to get at the assigned taxonomy at the genus level. I was lucky enough to have another graduate student, Sangam Kandel, to do the data analysis for me. And we actually got this data back just a few weeks ago. So it's really exciting. He took the data, the raw data, and imported to his software when he was able to do the filtering and assigning taxonomy of diversity in our abundance. The metrics that we use to look at diversity are alpha diversity or within sample diversity. We use the phylogenetic distance, which is qualitative to measure diversity. So it's based on the phylogenetic distance. More branches result in increased diversity. Beta diversity, however, is the diversity between communities or between our treatment groups. Again, this was based on phylogenetic distance with weighted unifrac. However, in our beta diversity, we wanted to take into account both the presence and the abundance of the specific bacteria. And to show the changes in relative abundance, we used taxonomic plots. And again, we went down to the genus level. For our AC immediate, we saw no changes in alpha diversity. So we have our faith PD values on the left, our treatment groups, and then the interquartile ranges represented in these plots. With our ACT immediate, we see an increase in alpha diversity that ends up declining after 1 month. Chemotherapy is known to decrease bacterial diversity. It was surprising to see this increase. However, it might take time to see this ending up decrease. For our beta diversity, we use a principal coordinates analysis to visualize the data. So if we look on the left for our ACT immediate group, this plot shows each individual sample. So we had 6 samples for our control, which are in the orange, and our 6 samples for our chemotherapy, which are in the blue. And what we're measuring for statistical significance here is whether the centers of these clusters are significantly distant from each other. The closer together that these clusters are, the more related they are in both abundance and type of bacteria they have. We do not see any significant difference with our ACT or AC immediate. However, when we look at our ACT immediate group, we see a slight spread between the treatment groups, and this ends up being statistically significant. However, when we look at our 1 month group, we see a further distinct clustering between our treatment groups, indicating a dramatic shift in taxonomy and abundance. So to look further into our taxonomy and the relative abundance, we have our taxonomic plots. Our relative abundance, the size of the stacked bar plot indicates the relative abundance. We had 6 samples, as I said before. On the left are our control samples, on the right are our AC. And each color represents a different genus, as put in the legend key over here. We see, as you can see, there's really no significant difference you can tell visually between the control and our AC animals. This makes sense in light of our alpha diversity. So we see no change in the alpha diversity or beta diversity. So we really wouldn't expect to see a significant change in the taxonomic plot. For our ACT immediate plot, we start to see some changes, however. Looking at our ACT treated mice, we see increases in the purple, which represent Myribaculaceae. And then we see an increase in the orange, which represents the genus Akkermansia. When we get to our 1 month post-chemotherapy, we see a dramatic shift in bacterial composition and abundance. In our control samples, the purple here represents the Myribaculaceae, which is present in the control. However, it dramatically decreases in our chemotherapy group. Then conversely, we see that we have lower amounts of the green, which is akkermansia here. However, that dramatically increases in our chemotherapy group. So a little bit more on what these genera are. Mirabaculaceae is actually an unified genus. It's actually unculturable, so all that we know from it is from metagenomic analysis. However, what we know from that is that it is a gram-negative bacteria similar to E. coli, and it is non-motile, it's mesophilic, and also does not sporulate. So you can get a lot of information just from the genome and from what enzymes are encoded in there. It was previously known as S24/7 or mouse intestinal bacteria because it's predominantly found in the mouse intestinal tract. It's found at a relatively high average abundance of 20 to 30%. And then I guess from functional analysis from the genome, they're predicted to be major utilizers of mucus-derived monosaccharides. An interesting point, I've started to look into, well, what does this bacterial do? Has it been seen in other studies? It has been shown to be decreased in polycystic ovarian syndrome patients. And then in a PCOS mouse model that's induced by letrozole, we also see a decrease in this genera. This is important because letrozole is one of the treatments in using for postmenopausal breast cancer patients. And what it does is inhibits estrogen production, so lowers estrogen levels. Chemotherapy by itself is known to induce medical menopause by lowering estrogen levels. So it's interesting that we see this connection. Next, akkermansia. Less is known about it. It's thought to be more of a beneficial. However, it's still being studied for its role in obesity, diabetes, and inflammation. It does have a few type species that have been analyzed. However, there are a lot of unclassified species here that need to be analyzed. It has been shown in studies that the abundance of akkermansia have correlated with anxiety and depressive-like behaviors. However, in these models, if socially stressed mice and of chronic restraint, they see these levels decrease. Where we see an increase with anxiety, we see an increase with akkermansia. To summarize, for our gut microbiota, we saw no significant differences between our gut microbiota and our AC immediate time group. Our ACT immediate, we saw a significant increase in alpha diversity and a significant difference in beta diversity, and that was represented by increases in akkermansia and Mirabaculaceae. 1 month after ACT treatment, we saw a decline in Mirabaculaceae and an increase in akkermansia. So to put this all together, in our AC immediate time group, we saw no changes in anxiety or depression. We saw a decrease in crypt depth and villus height, and we saw a decrease we saw no significant changes in the gut microbiota. And then our ACT immediate group, we saw no affective behaviors again. We saw decreases in villus height, and we also saw significantly increased diversity. Our ACT 1 month, we saw our increased anxiety, our decrease in crypt depth, and we also saw significantly altered gut microbiota. Next, I'd like to switch gears onto more of the cognitive side of things. Normally, other neuroseminars, the Allen lab gets asked, like, have you looked at other behaviors? And I looked at affective behaviors in my previous experiment. However, we mainly look at the effective or the negative effects of chemotherapy on cognition in mice. And this is due to chemotherapy-induced cognitive impairment. This phenomenon is now a recognized clinical syndrome characterized by memory, learning, and motor function impairment. It has defects in all these following domains: working memory, executive function, attention, and processing speed. The prevalence is hard to quantify. However, it's estimated that up to 75% of patients experience these deficits during chemotherapy. However, a subset of patients continue to experience these deficits after treatment, even months to years afterward. The colloquial name for this is chemofog or chemobrain, which is what I'll be referring to this syndrome for the rest of the talk as. The symptoms are very heterogeneous between each patient, and it can range from mild to moderate cognitive impairment. Talking to some breast cancer survivors myself, one woman suggested that it felt like her brain had aged. And then a family friend of mine, she just says it's hard for words to come to mind. And other studies looking at this have shown that women, this have a negative impact on their life since they don't feel as confident or in their ability to go back to the workforce, or they feel like they're never able to go back to they're at a new normal and they're not going to be able to go to their pre-cognitive state. before breast cancer. One of the potential mechanisms around chemobrain is oxidative stress. Oxidative stress is the imbalance between the production and accumulation of reactive oxygen species and the cell's ability to deal with that. Some consequences of oxidative stress is they can modify proteins, lipids, and DNA, all affecting their function. They can induce mitochondrial dysfunction, which further adds to the oxidative stress. They affect the endoplasmic reticulum. They can cause ER stress, which leads to protein aggregates. RNase metabolism also happens. We get impaired axonal transport in neurons. And if the cell reaches a threshold to where it can no longer handle this oxidative stress, it will undergo cell death. The brain is particularly susceptible to oxidative stress due to its high lipid content. It also consumes 20% of the body's total oxygen. And compared to other tissues like the liver, it has a modest antioxidant defense. It has a lower catalase content and, for example, lower glutathione content. So considering that chemobrain is a potential mechanism, we start to think about, well, what are some possible therapeutic options to treat this? Because as of right now, there are no FDA-approved drugs to treat chemobrain. This is where our redox modifier of interest comes into play. This is a really interesting molecule. It's a manganese porphyrin that was developed as a mimetic of the human mitochondrial manganese superoxide dismutase, or MNSOD. It catalyzes the superoxide radical into oxygen and hydrogen peroxide. This is a third generation molecule. It's both cationic and lipophilic. This lipophilic nature enables it to cross the blood-brain barrier, and it's able to protect against oxidative damage. In addition to protecting, let's see, normal tissue, this redox modifier has also been shown to sensitize tumor cells to both chemotherapy and radiation. And also has a very safe toxicity profile, which results in it being in a couple clinical trials. This compound right here, MNBOE, is known as BMX-010. Its sister compound is BMX-001. BMX-001 is in clinical trials to protect against anal cancer, and then BMX-01 is actually used to protect normal tissue and head and neck cancer. And that leads to, we want to determine if our manganese porphyrin could ameliorate the effects of a common breast cancer chemotherapy on hippocampal dendrite morphology and cognition. Here's my experimental design. Again, we used the same age, same type of mice as my previous study, female, regular black mice, and at age 12 weeks. Again, we give our ACT chemotherapy regimen with our 4 cycles of AC and our 4 cycles of paclitaxel. We then give our redox modifier starting 24 hours after our last paclitaxel, and that is given twice a week, and this one is given subcutaneously for 4 weeks. So a month after our last injection, we can do our cognitive testing with Morris-Water maze. Where then, again, we use our same dosages as before, and then we have our dosage of our redox modifier below. We use the Morris-Water maze to measure hippocampal dependent spatial memory. And this is our training schedule as we've seen below. We have a large pool, a circular pool that's filled with water, and it's opaque, so the mice cannot see through it. We teach the mice where a platform is. We place the platform in the 1 of 4 quadrants, and where the platform is, it's known as the target quadrant. We have 5 days of training. The first 2 days are visible platform training. Our visible platform is slightly above the water, and we also add on additional visual cue, in our case, a Lego, to guide the mice to the platform. There are 3 trials in the morning and 3 trials in the afternoon. On the 3rd day, however, the 3rd through 5th days, we hide the platform. So we take off our visual cue, then we slightly submerge the platform under the water. And now the mice have to rely on their extra maze cues and their memory to find the platform. To test spatial memory retention, we do what is known as a probe trial. So after the last trial of at the end of the day, we take the platform out, and then we let the mice swim around for a full minute, and then we measure the amount of time they spend in each respective quadrant. We find that our mimetic was able to maintain spatial memory. Now, if we look at our first set of data for our day 3 probe trials, we have our percent time spent in the quadrant on the y-axis. We have our 4 treatment groups on the x-axis. And if we want to look at our saline animals, they show prototypical results of a normal mouse in the water maze. They spend significantly amount of time in the target quadrant, indicating intact spatial memory. We see that our chemotherapy animals, however, do not spend a significant amount of time in any one of the quadrants, indicating impaired spatial memory. Our manganese porphyrin itself did not impair spatial memory, but importantly, when we add the manganese porphyrin to our chemotherapy, we see that it keeps intact spatial memory. On day 4 of our probe trials, with additional training, we're able to see that our chemotherapy, we're able to learn where the platform is, and thus spend more time in the target quadrant in the probe trial. However, upon additional trainings on day 5, we see a decrease again in spatial memory in our chemotherapy group. However, if we look at all 3 days, we see that our manganese porphyrin, in addition to our chemotherapy, was able to maintain spatial intact memory. As I mentioned before, the Morris water maze is a hippocampal dependent test. And so what is the hippocampus? The hippocampus is a brain structure that's located in the medial temporal lobe. It plays a crucial role in learning and memory, also short-term to long-term memory consolidation. And for this test, it plays a role in spatial navigation. The 2 principal neurons of the hippocampus are the granule cells of the dentate gyrus, and then we have our pyramidal cells that are in the cornus and monas regions, or CA1. There's different parts. So the dentate gyrus is where neurogenesis takes place. And then for the trisynaptic circuit, or how information flows through the hippocampus, we get input into the dentate gyrus, then which go along the mossy fibers to the CA3 region. The CA3 then connects to the CA1 through the Schaefer collaterals, and the output goes back out through to the intercranial cortex. To look at dendritic morphology in the hippocampus, we did Golgi-Cox staining in order to visualize, there we go, so you can see a little bit more. The Golgi-Cox staining method randomly stains about 5% of the neurons, so we're able to identify the neurons, their axons, and their relative dendritic branches. And then we are able to measure, by using concentric circles going out from the cell body, we're able to measure the dendritic length as measured from the distance from the cell body. If we look at the dentate gyrus on the left, we see our dendritic length as a function of distance from the soma. Our saline is in black and our chemotherapy is in red. We see a significant decrease in dendritic length from 100 to 200 microns from the soma. When we look at the addition of our manganese porphyrin with our chemotherapy, we see that it's able to maintain the dendritic length. We also see the same pattern in our same results in our CA1 apical and our CA1 basal area. Again, chemotherapy significantly decreases dendritic length. However, when we add in our mimetic, we see that's able to maintain that length throughout the same region. And we see the same results in our CA1 basal. Next, another measure of we looked at was dendritic complexity. It is shown in Alzheimer's and other neurodegenerative diseases that impaired cognition is associated with decreased complexity. And not only takes into account the dendritic length, but also the different branch points and branch tip orders. We see in our CA1, both in the apical and basal, our chemotherapy in red, it significantly decreases dendritic complexity. However, when we add in our mimetic to our chemotherapy, it's able to maintain that in both the basal and CA1. So just to summarize, we saw that our chemotherapy treatment significantly decreased dendritic length in both the dentate gyrus and the CA1 regions of the hippocampus. We saw the addition of our manganese porphyrin was able to maintain this dendritic length. And then we saw that this porphyrin was also able to maintain dendritic complexity in the CA1. As part of this project, We took a subset of hippocampal lysates, and then we did label-free quantitative proteomics on this. And from these results, both looking at through network analysis and pathway analysis, we saw common pathways that are affected both in our chemotherapy group and our chemotherapy redox group. We saw mitochondrial function, oxidative stress, and energy metabolism as all potential pathways that are affected and might be ways about how our redox modifier is working. And some pilot data that supports this is we looked at mitochondrial function in our mice 6 months after our chemotherapy treatment. We did this by using the Seahorse flux analyzer. And what we're looking here is the respiratory control ratio. A higher RCR implies a higher capacity for substrate turnover, ATP production, and a low proton weight, all for healthier mitochondria. We see in our chemotherapy group, 6 months post-chemotherapy, we see a decreased RCR rate. However, when we add in aromatic, we see an increase relative to normal. So to conclude, we see that our redox modifier maintains intact spatial memory. We see that it maintains dendritic length in the DAG and CA1 regions. It maintains dendritic complexity in the apical and basal CA1. And then looking at our bioinformatics from our proteomic data revealed some mitochondrial related proteins and pathways that are to be affected by our chemotherapy and then our chemotherapy and redox modifier treatment. Now to addressing the mechanism of MNBOE, of how it works. Given that we use our initial rationale to use this in the first place was considering that oxidative stress was a potential mechanism behind chemobrain, we wanted to see what is its role exactly in the oxidative stress response. And a few studies looking at these manganese porphyrins in the most recent 2 to 3 years have shown that the transcription factor, nuclear factor, erythroid-2 related factor, or NRF2, is affected. For example, in hematopoietic stem cells, when manganese porphyrins were added, they actually enhanced the function via activating the NRF2 pathway. And in the normal prostate fibroblast, they are protected by their sister compound from radiation by activating the NRF2 pathway. So going into more of the NRF2 pathway, it is a master regulator of the antioxidant response. And it's just a diagram of the classical pathway is shown here. NRF2 is usually kept bound by 2 molecules of its repressor protein, KEAP1, and KEAP1 is bound to the ubiquitin 3 ligase colon 3. Under normal conditions, NRF2 is quickly degraded because it is ubiquitinated by NRF2. NRF2 is ubiquitinated by COL3, and then it's sent off for proteomal degradation. So when we add in our manganese porphyrin, it's shown that the manganese porphyrin oxidizes key residues on KEAP3, which disrupts this interaction, which releases NRF2 to be able to go into the nucleus and thus target or transcribe for the antioxidant response. So NRF2 protects against endogenous and exogenous stressors, such as reactive oxygen species caused by chemotherapy and radiation. So our goal into looking at the mechanism behind this redox modifier was to see if NRF2 plays a role by looking at NRF2 knockout mice. So we're just going to look at our wild type data first from our Morris water maze. We have, as we saw in our previous results, we see that chemotherapy significantly impairs spatial memory retention on day 1 or on the first probe trial. However, unlike before, we saw that upon additional training in the hidden trial days, hidden platform trials, that our chemotherapy mice were able to maintain intact spatial memory. Looking at our knockout mice, NRF2 knockout mice, we see that most importantly, our knockout mice in the control group maintained intact spatial memory throughout all 5 days or all 3 days of the probe trials. We saw that the addition of our redox modifier also did not affect spatial memory. However, when we give our chemotherapy, it continuously shows deficits in spatial memory retention all 3 days. And importantly, when we add in our redox modifier, in addition to our chemotherapy, we do not see any maintenance of spatial memory retention. So this inability of the manganese porphyrin to protect spatial memory retention in our NRF2 knockout mice suggests that NRF2 may play an essential role and the ameliorative effects of this manganese porphyrin on cognition. However, we do need to confirm this pathway in our wild type mice using Western blots, just to verify that we see an increase in NRF2 and an increase in their target genes. Further research into this, we can look at, the neurons are not alone. There's also other cell types in the nervous system, such as astrocytes, microglia, and oligodendrocytes. So it'll be interesting to see what chemotherapy has, how it affects these cells, and then how does it interact with the neurons. So just to summarize with a graphical summary, we have our chemotherapy that we give, breast cancer chemotherapy. After a month post-treatment, we see increased anxiety, which is correlated with our intestinal morphology changes and our gut microbiota changes. And we also see this 1 month time point, a decrease in cognitive performance. Potential pathways that can be used to explain this and the communication between the gut and the brain, as I mentioned before, is inflammation. Reactive oxygen species also play a role in the inflammation response. They can further feed forward for each other. But we have to also consider there's other possible mechanisms behind our manganese porphyrin. For example, nuclear factor kappa B is another redox sensitive transcription factor. And it's actually a key regulator of the inflammation response. We know that our manganese porphyrin oxidizes KEAP1, which then activates NRF2. We also know from previous results looking at the mechanisms that manganese porphyrins have been shown to oxidize the NF-kappa B inducer kinase, which ends up inhibiting NF-kappa B. NF-kappa B also reacts with KEAP1, and then NF-kappa B can also modulate NRF2 levels because there is a response element within the promoter region of NRF2 where NF-kappa B can control the transcription of NRF2. Here's my references. I would really like to acknowledge the Allen lab. We have quite a crew, and then the previous lab members, Tyler Alexander, Thomas Groves, Julie Anderson, and Fred Kiffer. They all trained me before they left, especially Fred. And then we have a whole host of undergraduate and high school students who I've got to had the pleasure of training over the years. As Dr. Allen said, we've had some fun times. My Turkish roommate, that was one better experience of grad school, had to like a Turkish sister. We had karaoke with our undergraduate summer students. Tarian we got to present our first talk at a conference together. We continually celebrate everyone's birthdays. And then I'd like to end with more acknowledgement, especially my advisory committee for helping me forward with this project. The Aiken Burns Lab, Kim, Francesca, Alev, we work closely together. And the Division of Radiation Health, VG and SUDIP. I'd also like to thank the departments of neurology and biochemistry. I feel truly interdisciplinary. Y'all both made me feel welcome. The Student Wellness Center for helping me with my anxiety and depression throughout grad school. Of course, our DLAM and our other collaborators. A big shout out to Dr. Oberly Deegan. She is a real MVP because she provided me with the manganese porphyrin, and she was able to get the last batch out to me for my studies right before COVID hit. So we were able to continue on with this project. Of course, our COBRA's Center for Translational Neuroscience and the Centers for Studies of Post-Response to Cancer Chemotherapy. A big thank you to Dr. Mike Robeson and Sangam for doing our data analysis. And then of course the cores here on campus, which helped us with our histology and our proteomics and our funding. We couldn't do anything without our funding. And a last thank you to the mice. We wouldn't be here without you. I'd like to open up the floor for questions and the chat.
Thank you, Taylor. Wonderful presentation, and I want to congratulate you on a body of work. There are, I'll turn this over to Dr. Allen to moderate, the discussion. There are a number of, or a few questions from the chat as well. Gerard Denton had posted some questions, so make sure we sort of give him a chance to ask those as well. Dr. Allen?
All right. So Dr. Denton has several questions in the chat. Is there one or two in particular that you find more interesting, Dr. Denton?
No, just take your pick.
Okay. All right. Within the chat, one of the questions, Robin, is that do the immune cells infiltrate the CNS, or is inflammation mediated by cytokines?
We see, so chemotherapy has been shown to increase inflammation, both by pro-inflammatory cytokines, but then we do see increases in inflammatory cells in the CNS, but mainly the resident cells of the CNS are the microglia. So we do see those activated.
All right, second question. Do anxiety and depressive behaviors originate in different regions of the brain?
Okay, yeah, so different parts of the brain, especially the amygdala, and then we have the hypothalamic pituitary axis, adrenal axis, which can all play into a role in anxiety and depression. We have not looked at the amygdala part yet. However, that is something we want to do in the future. I did not mention this, but in addition to all the microbiota studies, we did collect hippocampal samples for staining so we can quantify dendritic complexity there.
Great. Yeah, I'm trying to understand how you could get changes in anxiety behavior and not depressive behavior.
Well, there's also other tests for depression that we could use, such as the sucrose preference test, which will test more for like anhedonia rather than the tail suspension and forced swim are good for like knockouts and antidepressants. We were just wanting to see if chemotherapy would cause depressive behavior in that. But there are other avenues to look to verify depressive behaviors.
Thank you. Another question I had is whether or not you have measured, so you mentioned early on that there might be a component of estrogen signaling in your cognitive impairments.
Yes.
Have you measured estrogen in your AC models?
We have not with mine. However, we work also with radiation and some NASA projects. We are just now getting into measuring the estrous cycles of the mice. And that is definitely something we want to look to in the future, given that we see this medical menopause in both mice and humans.
And I guess the last question I would ask is whether or not these NRF2 knockout mice are sort of hyperinflammatory compared to wild-type mice. You characterize NRF2 as a master regulator of the antioxidant response. And so in the absence of that, are these animals sort of experiencing hyperinflammation? And
that we have not looked into, but that is why we definitely want to assess NF-kappa B, because it's suggested that in our NRF2 knockout mouse, we see a hyperactivation of NF-kappa B and thus inflammation.
Great, thank you. I'll shut up now.
Oh, you're fine.
All right, next.
I did have a question related to the depression and anxiety tests that are available for rodents, and what other options are there? So the depression tests, for example, are kind of responses to threat, and it's probably a little different than what the women experience when they're having breast cancer and chemopog. So I was wondering if there, I'm not that familiar with these tests, but are there things that might more closely parallel the anxiety and depression that the cancer patients would experience?
As I mentioned, there are other forms of anxiety-induced models, such as the chronic restraint or social defeat. You can also cue fear responses in that, so you can see how they respond to fear. However, most of the tests for anxiety have been with just looking at the exploratory behavior of the mice, and then there's also a obsessive compulsion in the Marble-Berrian test that we can look at.
Mm-hmm. Fantastic. And in terms of the other cognitive models that you use, are there tests of attention, or that might be something you can utilize here?
Yes, more looking at the prefrontal cortex, you mean, higher cognition. I would like to use the puzzle box. So measuring their escape times, giving different obstacles. We also have the option of using an IntelliCage to continuously monitor the animals. However, we mainly stick with the hippocampal test because preclinical models of chemo brain have shown that it's the hippocampal dependent tasks that are chronically impaired on the long term.
Thank you.
Okay.
Oh,
If there are no more questions,
I have a
Um,
question.
oh.
Of course. Now we have a few minutes left. Let's push her.
yeah.
So Taylor, I remember reading in one of your papers, I believe earlier this year, where you did proteomics and looked for proteins that are up and down regulated in
Yes.
response to chemotherapeutic agents. Is that correct? Do I remember that correctly?
Yes.
Okay. I remember that there was a component of calcium channels or calcium signaling molecules in one of the data sets.
Yes, we saw that a calcium channel, a specific calcium channel protein was downregulated after chemotherapy. And I do understand that there are, like in psychiatric disorders, there's different mutations in these things where we see psychiatric disease occur. So it is, given the importance of calcium signaling and homeostasis in the brain, that is something that we should probably look at as at a functional level, in addition to just
Yeah,
the neurological changes.
You know, they play really critical roles in synaptic activity processing. Has anyone ever looked with EEG at your mouse models? See what happens, sort of cortical activity in response to chemotherapy?
Not that I'm aware of. There has been like computer simulations in silico done, and it's estimated that it does. We hypothesize that it does affect it, but we have not looked at it.
Gotcha. Okay, thank you. Now I'm really done.
Oh, you're fine. Thank you, Dr. Ditton.
Well, if there aren't any other questions, I just wanted to mention that you had 72 attendees, which is a great response for our seminar. It might actually be the record. I didn't keep track and I didn't count everyone's, but
Thank you
certainly
everyone for coming and supporting me. That means a lot.
did a nice job. Thank
Good
you.
job. Good job.
Thank you, Dr. Childs.
all right, guys. Thank you guys for attending, and we'll move on to the committee part next. You guys should have the link.
Great job, Taylor.
Thank you.
So, Dr. Allen, are we going to just stay on this meeting? Is that the plan?
Robin, did you send Dr. Minnemeyer the committee link?
I did not, but let me do that real quick.